All right, why don't we get started? Again, welcome you to this webinar series on human islet isolation. I'll give Bala an introduction shortly, but I just want to let you know a few ground rules before we get started. There are 3 polls, 1 poll at the beginning and 2 polls at the end. If you just take time and answer a few questions, it would be very helpful to us to see how this benefits you. There will be a Q&A session at the end. Please use the chat box to write down your questions, and I'll review them with Bala at the end of the webinar. In terms of this whole series, we're trying to keep a quarterly schedule, but it's difficult at times to find speakers. So if you have a topic that you think would be interesting for me to recruit a speaker, or if you want to present in one of these webinar series, please contact me. And lastly, we are going to be beginning a new webinar series. We held a workshop at the Joint Cellular Transplant Congress last October, and I announced this. It's going to be a webinar series with special topics in cell isolation. It will be topics outside of islets or human islets. And on May 16th, Rafe Wittek, who has got extensive industry experience in isolating primary hepatocytes from all species, including humans, will be giving a webinar on isolating primary hepatocytes, overview tips and tricks. So without any further delay, I want to welcome Bala, who's giving another webinar on which we're very pleased with, on defining islet isolation success in chronic pancreatitis pancreas and brain dead donor pancreas intended for auto and allo transplant. As many of you know, Bala has extensive experience in all sorts of islet isolation techniques. He's been doing it for over 27 years and has over 200 publication and serves on multiple editorial boards. So without any further ado, Bala can start the slideshow, and I'll make sure this first slide goes up.
Yeah, thanks, Bob.
So we need this slide, just get the slideshow. Okay, I see your thumbnails, and yep, it's up and running. So
okay,
we'll mute myself and turn my video off.
Thanks, Bob. Good morning, everyone. I hope you can hear me well. Bob, can you hear me?
Yes, I can, Bob.
Okay, fantastic. Yeah. Good morning, everyone. Thanks for this introduction and for this opportunity to talk about technical aspects of human islet isolation for defining islet isolation success for auto and allo transplantation. Isolating large quantities of high-quality islets from every resected pancreas is absolutely necessary for a successful islet cell auto and allo transplantation program. So during this talk, I tried to define islet isolation success based on the technical aspects for brain dead donor pancreas and chronic pancreatitis pancreas. This is primarily based on extensive literature available from our friends and colleagues over the period of several years, and my own experience in performing large number of brain dead donor pancreatic islet isolations for allo-transplantation as well as for basic diabetes research, and chronic pancreatitis pancreas for clinical auto-transplantation, and porcine and non-human primate isolations for xenotransplantation studies. These large animal pancreatic islet isolation helped us to define, modify our islet isolation technology. And interestingly, the split lobe islet isolation experiments, the specialty of this trisected pancreas model, in which we can learn a lot. Actually, we learned a lot from this trisected pancreas model, because we have a control lobe and two experimental lobes, because it's from the same pancreas. It provides a lot of newer data when we isolate the islet cells from this trisected pancreas model. The only thing is, it's a time-consuming process. It is almost processing 3 human pancreas in a day. I think at this, I really want to thank all my team members in the past and the present team members who have worked with me, participated, and contributed for all these islet isolation. As you know, islet isolation is a teamwork. I cannot do it alone. Once again, really thanks to all my team members. The specialty of this trisected pancreas model was easy for us to test various enzyme combinations in one pancreas. We used a 250 ml Riccardi chamber, 175 ml enzyme solution, and we alter the lobe for every isolation. So for each group, we had 3 pancreas to evaluate the result. And the experimental groups were blinded, so we don't know what kind of enzymes we are using during this islet isolation process. For example, we tested different proteases to see how those proteases cleave the peri-islet basement membrane to release islet cells without damaging islets. For example, Roche MTF comes with the thermolysin. What is the effect of thermolysin in comparison with the wider-side BP protease or servo-neutral protease? So in one pancreas, you get the actual result. We carefully monitor the pancreas digestion profile, including digestion time, gross morphology of exocrine cells, islet morphology, whether the islets are embedded or fragmented, and what was the cell pellet volume, whether the cell pellets were loosened type of pellet or compact pellet. So it's easier for us to learn many parameters with this trisected pancreas model. And we tested extreme doses of collagenase. In our mind, we wanted to know, is it possible to really kill the islet cells with the extreme dose of collagenase or extreme dose of protease? The common collagenase doses less than 21 unit per gram pancreas is sufficient to digest the pancreas. But in our experiments, we used the extreme dose of even 80 to 100 unit per gram pancreas to see what is the real effect on islet gross morphology. Not only during the islet isolation, we closely followed the islet function after culture. We did extensive quality control assessments to see whether the enzymes really affected or not. We tested cladribine as a protease. Instead of using protease, if you use cladribine alone, basically cladribine showed some special effect in cleaving the islet cells and giving a good islet morphology and all. So we tested in combination with the protease or without protease. In initial different protease enzyme combination testings, we used the same collagenase. It's all the collagenase from the Clostridium histolyticum bacteria, but the different proteases comes from different organisms. We also tested different ratios of C1 collagenase to C2. The common dose is 60-40 ratio. So if we alter the ratio to 40-60 or 70-30, what was the effect on islet pancreas digestion profile? And we also tested standard collagenase and increased protease, or low collagenase and increased protease, or the other way around, increased collagenase and low protease, to see what kind of effect we see in terms of islet morphology because protease plays a major role in digesting the peri-islet basement membrane. Actually, this experiment helped us to define the young donor islet isolation protocol. Based on this experiment, we commonly use low amount of collagenase and a higher amount of protease for younger donors. Interestingly, during islet isolation, we see quick release of islet cells, complete digestion of pancreas within few minutes when the pancreas in Riccardi chamber. So immediately we thought that maybe the enzymes which we use for this particular experiment was very high, because maybe the enzyme is very hot. That's why we see quicker digestion in this particular pancreas, because the experiments are blinded, so we don't know what dose or what enzyme we used. But interestingly, later when we analyze the data, we see a similar pattern of quick release even from the control lobe. So pancreas characteristics play a major role in releasing the islet cells, or digestion time, or pellet volume, or islet morphology, exocrine cell morphology, and all. So we all use a semi-automated method developed by Dr. Camillo Riccardi and his team. And the basic method of islet isolation is intraductal injection of enzyme collagenase to digest the pancreas in Riccardi chamber, followed by purification using density gradients using COBE2991 cell processor. The fundamental problem in islet isolation is a large variation in islet yield. As you see here, you can see large different number of islet cells from different pancreas. This graph, this data from a multi-center study analyzed more than 1,000 human pancreas. The data was put together by Linja, and it clearly showed a lot of variation. Then historically, the same phenomenon was reported by Edmonton group in 153 consecutive islet isolation, and historically enzyme liberase HI was used. So we could see different islet number in historical time. Then the same Edmonton group reported another 185 islet isolation using all the modern enzyme, basically GMP grade purified type of enzymes. So even after experiencing more than 1,800 human islet isolation, it's difficult to control these islet isolation variables. Many papers are available on this topic to describe how donor characteristics and islet isolation characteristics predicting the islet yield. So these papers are from our friends and colleagues. And in their analysis, many different factors showed positive correlation or negative correlation in terms of islet yield. Collaborative islet transplantation registry also analyzed similar analysis using more than 1,000 human islet isolations, and published what are the different factors affecting the islet yield, or is it possible to predict the islet yield so that we can select the right donor for our recipient and all. To summarize, various factors, primarily age, height, weight, and BMI, or body surface area is a better marker than BMI. The weight of the donor plays a major role. Then cause of death, medical social history, biochemical values affect the islet yield, and the pancreas procurement technique, cold ischemia, and donor management also affect the islet isolation outcome. In general, the pancreas procurement technique or cold ischemia plus donor management can be kind of controllable. The effect of donor cause of death or medical social history or biochemical values also it affects in a milder way. But the important factors are donor weight and age. Those two factors played a major role in getting higher number of islet yield, or they predict the islet yield in all kind of analysis. Then there are some other factors negatively correlated. For example, anoxia as a cause of death was a negative correlation, and elevated sodium and potassium, they had an opposite effect. When we accept the donor pancreas, we don't get perfect donor score for all those pancreases. There are always some red flags. Sometimes creatinine levels are elevated. So if creatinine levels are high, it's a kind of indication of low blood flow to the pancreas. So generally we try to avoid utilizing high creatinine donor organs. Similarly, AST, ALT elevation, indication of some kind of downtime. And interestingly, even from a short cold ischemia, also kind of negatively affect negatively affected in terms of embedded islets. For example, if pancreas is from our local hospital, we are very excited. If pancreas is from our local area, we get the pancreas within 2 to 3 hours, but we end up seeing a lot of embedded islets. It's maybe due to our hypothesis is pancreas is full of UW or HTK during the cross-clamp time, and UW doesn't contain calcium. Maybe during enzyme distension, it causes some kind of inhibition for collagenase activity, and we see a lot of embedded islets. Plus donor management with arginine, vasopressin showed positive correlation. During human islet isolation for brain dead donor pancreas or chronic pancreatitis pancreas, we see completely two different issues. During brain dead donor islet isolation for allo-transplant, many times we see low islet number at digest level, even before purification. So the procedure went very well. For example, the pancreas was accepted for clinical allo-transplant, cold ischemia was normal, and the procurement was done very well. Even then, using our standard procedure, pancreas was digested, good digestion, but the number of islets at that stage was kind of low. And sometimes we see large number of islets at the digest level, but if the islets are embedded, then we are having a lot of purification issues to recover those islets. That leads to, again, failed islet isolation. But during processing CP pancreas, we see a different type of issues. For example, digesting the CP pancreas itself is a challenge. And in many labs, unpurified islets are transplanted. There is no purification steps. So if pancreas from mild or moderate fibrotic cases, if we see large tissue volume of 30-40 cc, it is very difficult to infuse those islet cells without purification. During clinical islet allo-transplantation, we expect more than 350,000 islets at least, because in order to meet greater than 5,000 islet equivalent per kilogram body weight, at least we have a definition. We want to achieve this number at the time of clinical allo-transplant. But when we process pancreas for our research, we at least expect 150,000 to 200,000 islet equivalents per pancreas. But in the case of chronic pancreatitis, there is no real definition. Whatever the number comes, we transplant, and we believe that's the best number for that particular pancreas. So we have to define what is the islet isolation success for chronic pancreatitis pancreas. In these two cases, brain dead donor islet isolations, 50% of the time, we don't achieve this number, our expected over 350,000 or over 150,000 number. 50% of the time, we don't, and it caused a lot of issues in terms of utilizing those islet cells, not for transplant purpose or even for the research purposes. The best example is the data from clinical islet transplantation consortium data. this manufacturing data analysis showed from 324 human islet isolations using standard unified donor acceptance criteria, unified SOPs and batch production record, and even after using all modern enzymes, purified enzyme, GMP grade enzymes, both collagenase and proteases are in separate vials. So even after using all these things, the successful transplanted islet preparations were only 52%. So it is important to analyze what are the factors causing successful islet isolation and what are the factors really causing failed isolation. This is more important than this aspect. So in the extensive analysis of successful islet isolation and failed islet isolation, we noticed significant decrease in pancreas weight in failed isolation compared to successfully transplanted preparation. I think among different donor characteristics and islet isolation characteristics, pancreas weight plays a major role. Age, height and weight really affect the pancreas volume. We see a lot of evidence in the literature, increased weight, how it really affected the pancreas volume. So evidently, increased pancreas volume means more number of islets in the pancreas, plus changes in the extracellular matrix. When pancreas volume is more, especially from higher body surface area donors or higher BMI. High BMI is somewhat tricky, so the best assessment is higher body surface area donors. Edmonton group reported a nice paper on this topic. It is even calculate how much is our pancreas weight based on the body surface area. So those factors increases the pancreas weight. So this entire pancreas provides necessary insulin for metabolic function and exocrine secretions for digestive functions. So there is a lot of changes in the extracellular matrix. That's what we see it in islet area fraction, histomorphometry, islet pathology studies. The Peter Butler group reported that the fractional pancreatic insulin positive area, pancreas parenchymal area, average islet cell, and average islet density are all moderately increased in higher weight patients compared to lean donors. For example, here, significantly higher amount of pancreatic parenchymal volume, beta cell mass, and beta cell area were observed in obese donor compared to lean donor. Similar observation was noticed in other study. Donor body weight was directly correlating with pancreas weight. Not only pancreas weight, the other factors, extracellular matrix changes or fatty infiltration in the pancreas, and there are unknown factors definitely causing nice islet release from those heavyweight donors. For example, whenever we isolate islet cells from a firm fatty pancreas, we see a nice free cleaved islets when compared to soft pancreas with the jelly capsule. This is the way we see from a lean donor. We see some islet cells. Mostly they are all embedded, and this is the way we kind of see from a fatty donor, firm donor. So these extracellular matrix changes definitely allow the enzyme to release these type of islets when compared to islet release from lean donors. So when we analyze the data from these failed isolation, I think this is the main focus for us, how to really avoid these failed isolations. Islet area fraction studies and histomorphometry studies and histopathological studies, plus assessing how much tissue pellet we get from 1 gram pancreas. For example, generally 1 gram pancreas gives 0.3 to 0.4 cc tissue pellet, plus failed isolation, all kind of univariant and multivariant analysis from failed groups, and even from the historical islet isolation when we used the Liberase HI from the failed group. I think it's important to kind of select the right donor in order to reduce our failure rate. In general, if we get greater than 3000 islet equivalent per gram pancreas in 0.3 cc per gram pancreas, in my view, it is considered as a successful islet isolation. In physiology textbook, it says every pancreas contains 1 to 2 million islets, but definitely we don't see in many pancreases. There is no proper even reference for those 1 to 2 million islets. Dr. Oli Koskren historically showed there is not many islets in some pancreases, especially in the lean donors. Now, before I switch to chronic pancreatitis pancreas islet isolation, I'm missing one slide. So at this point, all different analysis clearly showed increased pancreas weight can really release the islets in an easy way for enzyme to really act on the peri-islet basement membrane to release the islet cells. So we get free islets as well as large number of islet cells. So we have to really focus on selecting the donor based on our recipient islet requirements. Now I switch the topic to chronic pancreatitis islet isolation. As you see here, isolating islet cells from chronic pancreatitis pancreas poses multiple challenges: alteration in ductal structure, calcification in duct, and accumulation of fibrous layer in the pancreatic parenchyma can significantly hinder the ability to inject the tissue dissociation enzyme. If you compare the gross morphology of brain-dead donor pancreas and the chronic pancreatitis pancreas, you could appreciate the difference. There are many differences between CP pancreas and brain-dead donor pancreas. Primarily, CP pancreas coming from a living donor, and brain-dead donor pancreas, they are exposed to cytokine storm. And in general, the cold ischemia is very kind of low for CP cases compared to brain-dead donor pancreas. And one important aspect is the blood content is generally very high in pancreatitis pancreas compared to brain-dead donor pancreas. Because in brain-dead donor pancreas, there is a cross clamp and a flush of organ preservation solution to all the organs to reduce blood clot in pancreases. Histologically, we see large variation in percentage parenchymal cell. For example, there are some islets, but mostly fibrous tissue. The parenchymal cell percentage is different in histological studies. This is a normal pancreas. It's the entire pancreas is covered with a lot of parenchymal cells, including islet and acinar cells. Sometimes this is a normal islet morphology. Sometimes we see a lot of variation or changes in islet gross morphology. And the ductal anatomy also different. Sometimes we see large dilated duct or or sometimes calcification in the duct, or sometimes we don't even see the duct at all. So this data from University of Minnesota showed that transplanted islet mass varied from less than 5,000 to over 1 million islets from consecutive 476 isolation. In general, acinar atrophy and the degree of fibrosis affect the islet yield. When pancreas comes to the lab, we categorize them into mild, moderate, and severe cases. If pancreas is within these two categories, either minimal change or moderate, generally we don't have much problem in digesting these type of pancreas. The main focus is how to really digest the severely changed pancreas. The minimal change pancreatitis gross morphology is similar to brain dead donor pancreas, and we get comparable islet yield from this minimal change chronic pancreatitis cases compared to brain dead donor organs. Generally, modified islet isolation steps are necessary for digesting these type of severely fibrotic cases. For example, we need to go with a higher dose of collagenase, and parenchymal injection is necessary. Enzyme recirculation is necessary. Prolonged enzyme distension steps are necessary, but though these steps are not necessary for our brain dead donor islet isolation. When we see low number of islet cells in chronic pancreatitis pancreas, there are two possible reasons. Maybe the number of islet cells in the pancreas is low, or there is a technical difficulty to recover islet cells. When we analyzed chronic pancreatitis pancreas histology and islet area fraction, sometimes we see a large number of islet cells. For example, the islet area fraction for score for this pancreas is 4.5, but it's under severe category. So it's it's predominantly there are a lot of islet cells because it's a chronic pancreatitis is an injury to the pancreas. That injury stimulates a lot of hypertrophy of the islet cells and all in many cases. So I think primarily we deal with the technical difficulty to recover islet cells. In chronic pancreatitis pancreas, after islet isolation, we see large amount of undigested pancreas. So this is our focus to minimize this undigested portion of the pancreas, because we lose a lot of islets in these undigested pancreases. So in my opinion, the percentage undigested pancreas should be the cutoff for islet isolation success in CP cases. It is possible to achieve good digestion to minimize the undigestion of these chronic pancreatitis pancreases. For example, it is important to deliver the enzyme through both ductal as well as parenchymal injection approach. And we need to assess the percentage parenchymal cell before we select the dose of the enzyme and the selection of collagenase. So this assessment will help us to really identify what percentage of parenchymal cells we have from every resected pancreas. And based on that, we have to make sure we have the right amount of collagenase to digest these pancreas. We don't want to simply use just one vial approach. Sometimes the one vial approach enzyme dose may not sufficient to digest this type of pancreases. This is the classical example from brain dead donor pancreas, undigested tissue mass. And this is a chronic, good example of a chronic pancreatitis pancreas, undigested tissue mass. And this is the bad example of large undigested tissue pancreas. And histologically, from these samples, we lose a lot of islet cells. So we want to minimize to this level or this level. As you know, enzyme distension is the critical step in the entire islet isolation process. We make sure to deliver the enzyme through the pump system or hand syringe injection, or automated pump systems to deliver the enzyme. Sometimes we need to inject the enzyme with the help of needle throughout the pancreas. For example, there is a large amount of whitish fibrous tissue in these cases. So we have to deliver the enzyme for a prolonged period of time in order to digest these pancreas. And cutting the pancreases into tiny pieces will also help help for the exposure of the so pancreatic tissue to the enzyme will also help to digest the pancreas. And many times we use this enzyme recirculation approach. In classic standard Riccardi procedure, we digest the pancreas, and once we stop the digestion, we start collecting the cells with the cold solution. In the enzyme recirculation approach, we open up the chamber, we open up the system, and start collecting the islet cells and add the enzyme back to the circuit. So this approach helps to digest the undigested portion of the pancreas. It's a simple recirculation approach. Whenever we see prolonged digestion of more than 30 minutes, we don't want to stop the digestion. We start collecting the released cells and quickly spin the conical and add the enzyme back to the circuit. At the same time, collect the islet pellet into the good medium to save them. Then when there is the type of collagen matrix in brain dead donor pancreas is very different from chronic pancreatitis cases. As you see here, there's a uniform quantity of collagen 1, collagen 4, collagen 5, and 6 are expressed in normal pancreas. But in chronic CP cases, these components are expressed in very high amount, sometimes more than 1,000-fold. One example is we have to make sure our enzyme is capable of digesting this laminin type 6 collagen, because we see these type of ECM matrix in CP cases. This data shows when we used intact C1 collagenase in combination with neutral protease, we achieved more than 90% digestion and less undigested tissue mass with a combination of dosing the enzyme, best delivery of the enzyme, and other factors like supplementing other protease to digest those pancreas to achieve this high undigested minimum undigestion of the pancreas. So I'm sure different labs they use different enzymes from different vendors. For example, but in all the companies they supply collagenase from Clostridium histolyticum. So it is important to use greater than 26 Wunsch unit per gram of pancreas for this chronic, for severely fibrotic cases. If your lab is using neutral protease, it's important to use more than 1.5 DMC unit per gram pancreas. If you are using this space, definitely it requires more than 20,000 space unit per gram pancreas. Thermolysin also, definitely it requires more for chronic pancreatitis islet isolation. Again, this formula changes depending upon the percentage pancreatic parenchymal mass. For example, if pancreas comes with only 20% of parenchymal cell, generally we can reduce the BP protease, generally we can reduce the protease component. We need more collagenase to digest the collagen fiber, because even if islets are embedded, it is good for transplant purpose, because we are not going to get large amount of tissue pellet from those cases. So depending upon the percentage of parenchymal mass, it is best to design the collagenase dose or any protease dose. We have successfully introduced recombinant collagenase for our brain-dead donor islet isolation, as well as for our clinical auto-transplantation, pediatric auto-transplantation and all. Historically, we tested in split lobe experiments. We tested the quality of the islet cells, even scanning electron microscopy of the islet morphology and all. So this recombinant collagenase, it's RC1 and RC2. It's a powerful enzyme compared to natural collagenase. So this enzyme, in combination with PP protease, it reduces minimum undigested tissue mass. So these are the modified approaches we commonly use for our CP cases. As I said, the first step is assessing the percentage parenchymal tissue and fibrosis level and categorize them into mild, moderate, or severe cases, and dosing the collagenase and protease based on the fibrosis assessment. For example, predominantly we need more of collagenase, less of protease for CP cases, but for brain-dead donor pancreas, we need a free islets, so we need to use more protease, less collagenase, and deliver the enzyme collagenase. This is the very tricky part, because assessing the distension quality is very important. We try through the ductal first, and parenchymal injection many times is required for severe fibrotic cases. Generally, it doesn't require for mild and moderate cases. And the enzyme recirculation also enhance the undigested portion of the pancreas. Generally, our islet isolation solutions contain cytoprotective compounds in recombination and collection flasks that reduces stress for islet cells. And the last aspect is we try to reduce the exocrine cell mass with the modified purification procedure. This is the way we want at the end of CP case islet isolation. In my view, less than 15% of undigested pancreas is somewhat a measure of islet isolation in CP pancreas cases. Very briefly, in 1-2 slides, I would like to explain about the young donor islet isolation, because we do many pediatric islet auto-transplantation. In young donor, we see mantle islets. It's very difficult to purify these mantle islets because the islets are not completely cleaved from the peri-islet basement membrane. There is an exocrine cell is always attached to the islet cells. So purification is impossible if we get the islets like this. And in University of Minnesota, Rita Bottino and our team developed this young donor islet isolation protocol. At the time, we used liberase HI enzyme. So we have to mimic our animal islet isolation protocol, basically static islet isolation protocol, instead of the standard Riccardi islet isolation method. So prolonged enzyme distension and placing 2 screens in Riccardi chamber and the extremely slow mechanical shaking helped us to get more mantle-free islet cells. Then, in the absence of liberase HI, using our trisected pancreas model, we learned more proteases necessary, less collagenases also important, so that we don't get large clusters of cells during islet isolation. So prolonged enzyme injection with less collagenase, more protease, and minimal mechanical shaking, these steps will help to get less mantle islets or more free islet cells. 2 papers published on this topic in young donor islet isolations. And then the last one slide, I will finish up with the purification approach. In standard purification process, we lose a lot of islet cells and we get pure islet cells with the smaller pellet volume. In CP cases, our approach is we use a high density purification approach. Our purity level is generally very low, but our goal is to really reduce the exocrine cell volume. The word purification is misleading in the case of CP cases. Our goal is to reduce the exocrine cell mass. We are not worried about the purity of the islet cells. In University of Minnesota, we developed this analytical test gradient system. Using this analytical test gradient system, when we tested exocrine cell density from chronic pancreatitis pancreas and brain dead donor pancreas, we could see a distinct difference in those two exocrine cells. Because autografts, it comes from a living donor with Charcot ischemia, so the exocrine cell densities are generally high. So if we use a standard densities, which we use for our normal pancreas, we lose a lot of islet cells. Most of the islets will be in the co-bag. So we have to use a higher density approach. Basically, if we use the higher density, it pushes the islet cells to upper layer, and generally the exocrine cells will contaminate the acinar cells, contaminate the islet cells, but we don't end up losing a lot of islet cells in the co-bag. This is our standard purification approach we follow for our brain dead donor pancreatic islet isolation. We get a pure islets, but most of the exocrine cells are in the bottom. So this approach definitely will help to get good recovery of islet cells. This data shows our initial digest pellet volume was 30 ml, and we reduced to 11 cc with the recovery of 87% with the higher density purification approach. To summarize today's talk, I think we need a new enzyme formulation for lean donor, because based on the ECM analysis, because the current enzyme formulation, it gives some kind of embedded type of islets from lean donor. We don't see a nice free cleaved islet cells. And definitely donor selection is necessary based on the islet equivalent requirement for the recipient. Because you know that your recipient body weight before starting the islet isolation. So if a recipient is 80 kilograms, at least you need more than 450,000 to 500,000 islet cells. So based on that, it is important to select the donor. Otherwise, we end up continue to fail in our islet isolation. And in my opinion, if we achieve over 3,000 islet equivalent per gram pancreas in 0.5 cc tissue pellet from a lean donor, it is a successful isolation. That's the kind of mark whether our technique is really correct or not. In the case of CP cases, again, assessing percentage parenchymal mass followed by modified islet isolation steps are necessary, and the high density purification approach will help to recover more islet cells. And in my view, if we see less than 15% of undigested pancreas mass, it's an indication of islet isolation success. Now, I thank our islet transplantation scientific community. I learned a lot from everyone. And organ procurement organization, they provided numerous pancreas for doing our research isolations. And I thank my mentors, team members, and funding agency. This is my current team. I thank my pancreas care team. With this, I stop. I'm happy to take any questions you have.
Yep. Thank you, Bala, for a very informative webinar. I mean, it's a difficult topic that you've because just looking at your slides, and unfortunately, a lot of the stuff that we try to make conclusions from are anecdotal observations.
Definitely.
And by the way, please feel free to write any questions you have in the chat. But and I mean, one of the examples that I think there's something to it is clostripane. And it isn't really mean it's just clostripane. It means it's a complementary enzyme. Actually probably helps. And, there's been people who, and it started with Daniel Brandhorst, who showed that the better lots of the Nordmark product had higher clostripane. For a while, clostripane was used and supplemented in Uppsala, and I don't know where it stands now. Just if you want to comment on it, supplementing with clostripane.
No, I think our observation, Bob, because our experiments were blinded. So whenever I see a good islet morphology, it was associated with higher TLA or supplementation of clostripane. Definitely clostripane is doing something, and it gives a better islet morphology. It's an additional way to cleave the islet cells from the peri-islet basement membrane. And yes, we are in the process of utilizing clostripane either as a separate protease or in combination with other proteases.
And one other question, there's a few questions that are now coming up. The same goes for calcium. There was a paper out of, again, Uppsala saying that you need to add more calcium. From our standpoint, knowing what we know of the biochemistry of these enzymes, as long as if you had 1 millimolar calcium, that's more than excess sufficient, which is what's in Hank's BSS. So I was just wondering if, from your this anecdotal observation, whether people modified the calcium.
Maybe for the last almost 15 years, we use plain Hank's medium. We never added extra calcium chloride to our enzyme solution.
Okay.
Our plain HBSS, definitely the amount of calcium in HBSS, Hank's balanced salt solution, it was sufficient to digest the pancreas.
Okay. So Stu Williams asked the question, great talk, Bala, a few questions. Tissue mincing, why not always cut the pancreas into smaller pieces? Agitation methods, anything better than marbles, endogenous enzyme inhibitors released during digestion? Should the digest be washed and new enzyme added? So the first one goes into mincing.
Definitely, Dr. Stu, I always like that approach because we are spending almost 30 to 40 minutes injecting the enzyme for the parenchymal injection with the needle. But the mincing approach will definitely help to digest the pancreas. Only thing is we don't know, what kind of islets we get after mincing the pancreas. It will be a quick process because many times I wonder because the longer enzyme distension may not be really good for the pancreas. And then your second question is...
Is agitation methods anything better than marbles?
I don't know. We have not tested.
You could use like a Miltani system or something, which just shakes.
And then the endogenous enzyme inhibitors released during digestion should be digest washed and then So Dr. Stu, answer to this question, we have used many different protease inhibitor, PEFA-block, for example. PEFA-block has been used for a longer period of time, and we explored different protease inhibitors. Definitely, exocrine cells will release a lot of endogenous proteases. That's the reason if pancreas comes with a longer cold ischemia time, sometimes we need a very less protease because the endogenous protease itself really help to digest the pancreas. But we didn't have a concrete data to really support whether this is really necessary or not. To answer to your question, actually, Memphis, University of Memphis approach, they did human isolation for a longer period of time. Dr. Osama Garber, they remove entire enzyme within few minutes after isolation,
really?
add a fresh enzyme and all. The same approach was followed in University of in Colorado, in Denver also. So we don't know whether that adding extra enzyme will really help or not.
Yeah, just to make a comment on the endogenous protease, there's a report that Jonathan Likey did around 2005 with, I think it was Rose was one of the authors on it, but they definitely showed that you could use Pefferblock to knock down the endogenous proteins. And there was an issue when they did another paper that 10 hours, I think it was, you'd only should use it when the cold ischemia time is over 10 hours. But then it's not been, again, one of these anecdotal observations, it's not been followed up systematically. All right, I'll go to Rita's question. Thanks for the great webinar. Do you have any experience with tips concerning isolations from donors that died of drug overdose?
The drug overdose donors are nowadays, it's very common. We continue to accept those pancreas. We don't reject. So in terms of any special tips for those type of pancreas, not really. So we follow the standard islet isolation procedure.
Okay. Greg Zott asked, nice comprehensive talk and congratulations on all this work. Do you know if the recombinant C1 versus recombinant C2 collagenase has different effects on different collagen species?
Maybe Bob can answer to
Yeah,
this.
that's what I was going to say. To the best of our knowledge, this gets into these, in general, C histolytic collagenase digests all forms. It also, when you, in our case, when you add this base, it's a good C4 collagenase type, I should say, a collagen type 4 collagenase. So we'll cut C4. So this question gets asked, and it's a difficult one to answer because it depends on, what's the substrate really in the matrix. But there is, I can tell you, with this base, it's used to isolate skin, so they know very well that it did digest it in skin.
Well, in our split lobe experiments, when we started using RC1 and RC2, even altering the ratio instead of 60-40, 50-50, or 70-30, we didn't see much effect.
No, you don't.
The role of C2 versus C1, we didn't see real benefit even if we altered the ratios.
Yeah, and I think just from what we're working with Jar Saccone, it's really they complement each other. And so you probably use a very wide range. And let's face it, for all those people who are using natural collagenase or crude natural collagenase at the beginning, those is 75 C1, 25 C2 ratios in most collagenases. Jose Cano asks, excellent review, Bala. What has been your experience with heavy alcoholic donor pancreas islet function?
Actually, it's poor. In CP cases and all, if alcoholic is the primary cause for CP, generally those pancreases are severely fibrotic, and evidently we don't get large number of islets from those alcoholic donors. and definitely the islet function will be poor. In normal pancreas or brain-dead donor pancreas, heavy alcoholic is always associated. It's a red flag. We will have a poor islet yield and a poor islet function.
Okay, so I think that's the end of the questions that I could see. I remind you to thank you first, Bala, for an excellent overview of the difficulties encountered in isolating islets from either chronic pancreatitis patients or brain-dead donors. I encourage you, before you sign off, to fill out the polls, and we will be scheduling another webinar. And if you have any interest in identifying a topic or want to present a webinar to the group, just feel free to contact me. Again, thank you all. Thank you, Bala.
Thank you all.
And I will sign off right now.